Assalamualaikum and hi everyone, I am Nur Hafiza. So, in this video, I will explain about the genetic materials that involve during cell division, which is chromatin, chromosome, chromatid, and also sister chromatids. Firstly, chromatin. Chromatin are thread-like structures that located inside the nucleus that made up of protein and deoxyribonucleic acid, or we known as DNA. So this is chromatin that contains centromere in the middle and it's not condensed yet. When the cell undergoing for cell division in process mitosis or meiosis, the chromatin will condense and become more thicker that called as chromosomes. As we all know, humans have 46 chromosomes, which is 23 pairs come from father and the other 23 pairs come from mother. Mitosis process at prophase, the chromosome will duplicate and become the chromatid. So this part we call as chromatid. And the centrioles here and also the spindle fibers. So this is chromosome and this part we call as chromatid. A chromatid is a one half of a duplicate chromosome. Next, sister chromatids. So this both we call as sister chromatid and also this sister chromatids. A sister chromatid is either one of the two chromatids of the same chromosome joined together by a common centromere. For example, during the anaphase, all the chromosomes will separate and the sister chromatids will move to the opposite end of the cell by the spindle fibers. And this is what we call as sister chromatids. Alright, that's all from me and thank you.
Hello everyone, my name is Audrey Elwana Joliti and now I will explain a few terms in cell division, which is homolog chromosome, tetrad chromosomes, centromere, and kinetochore microtubule. First is homolog chromosome. It is a pair of chromosomes made up of two homologs. Homolog chromosomes have corresponding DNA sequence and come from separate parents. One homolog comes from the mother and the other comes from the father. For example, humans have 46 chromosomes where one homolog is inherited from the mother and the other one from the father. The 22 pairs of homologous chromosomes are autosomal and the sex chromosomes X and Y make up the additional 23rd pair. Humans are diploid organisms because they contain two homologous chromosomal sets in each cell. Next is tetrad chromosomes. Specifically, when homologous chromosomes that have previously duplicated their DNA link up, only then it is called a tetrad. For example, during prophase 1, the homologous chromosomes condense and become visible as the X shape, pair up by synapses to form a tetrad, and exchange genetic material by crossing over. Moving on to centromere. Centromeres are the main chromosomal structure where proteins connect to spindle microtubules and facilitate chromosome segregation during cell division. For example, during metaphase 2, the centromeres of the paired chromatids align along the metaphase plate in both cells. For the kinetochore microtubule, the kinetochore assembles at the surface of the centromere and acts as the site of spindle microtubule binding during mitotic or meiotic division to pull the chromosomes apart. For example, in anaphase 2, the kinetochore on the outer surface of the centromere promotes attachment of the chromosome to spindle microtubules. When the chromosomes separate at the centromeres, the spindle microtubules pull the separated chromosomes toward each pole of the cell. That's all from me, thank you.
Hi, Assalamualaikum. My name is Iqlil Atisha binti Muhammad, and today I would like to explain about my spindle microtubule, haploid, diploid and somatic cell. At the beginning of nuclear division, which is meiosis and mitosis, centrioles position themselves at opposite ends of the cell, forming cell pole. Microtubule extend from central in all possible direction, forming what is called a spindle. For example, during anaphase, the chromosomes are simultaneously separated and pulled by the spindle to opposite pole of the cell. Haploid cell is a cell that contains a single set of chromosomes, which is the N is 23. Haploid cell, or known as sex cell, are formed through meiosis. The example of haploid cell is sperm and ovum. Both of these have 23 chromosomes in each cell. Diploid cell is a cell that contains two sets of chromosomes. This is why 2n equal to 46. As for diploid cell, it undergo mitosis, which is on my diagram here. In human, all other cells besides sex cell are diploid. This is the process of mitosis that produce two diploid cells. Next, somatic cell can be defined as a body cell. Each somatic cell has two sets of chromosomes, and they contain two copies of each chromosome, 23 from each parent. Somatic cells are produced through the process of mitosis, and this is why I'm using the diploid diagram just now. From the mother cell, it will produce two somatic cells, which is diploid cell, and somatic cell is very crucial because they make up various parts of the body, including all the internal organs, the connective tissues and bones. Thank you.
Hello everyone, my name is Amiruddin, and in this segment I will briefly explain about gamete cells, cytokinesis, karyokinesis, chiasmata and crossing over. So, gamete cells is an organism's reproductive cell. The example of gamete cells are sperm and ovum. Unlike somatic cells, gamete cells are haploids, which means that each cell carries only one copy of each chromosome. Gamete cells also produced through a type of cell division known as meiosis. Now let's move on to cytokinesis and karyokinesis. Cytokinesis is a physical process which divides the mother cells into two daughter cells. Cytokinesis is also involved in the formation of the cleavage furrow, which pinches the cells into two. This can be observed in the diagram here. Now, on to karyokinesis. Karyokinesis is a process where the division of nucleus occurs. Not to be confused with cytokinesis, karyokinesis divides the parental nucleus into two daughter nuclei. Now, karyokinesis is a continuous process. That means there is no pauses in between the process. Karyokinesis also can be divided in four major phases, which are prophase, metaphase, anaphase, and telophase. I hope that you're still with me because we are going to take a look at our last points, which are chiasmata and crossing over. There you go. Of all the stages occurred in meiosis, prophase 1 holds a particularly important as homologous chromosomes, which are one from paternal and one from maternal, paired and exchange their genetic materials. Here. This process is known as crossing over. This results in a recombinant chromosomes during sexual reproductions. The point of contact between two non-sister chromatids belonging to the homologous chromosomes, which are right here, is called the chiasmata.